Swayam Prabha. Digital India. Educated India.
In coming lectures, we are going to start a new topic, chromatography. Now, in chromatography, we are going to discuss about basic concepts of chromatography as well as some techniques, particularly the techniques of column chromatography. Now, as we all know that there are a lot of biochemical processes which are going on inside the cell. Now, to understand the biochemical processes, it is very important to understand the function and physical and chemical properties of the constituents or the active participants of these biochemical processes. And to understand their function or their physical or chemical nature, it is important to get these constituents in pure form, so that we can understand the particular function of an analyte or a particular molecule inside a cell. So, if we analyze, our understanding of biochemical processes have gone in parallel with our ability to isolate and purify biological molecules, which includes macromolecules such as proteins and nucleic acids, and also lot of other biological molecules. Now, most biochemical investigation requires they require that material may be purified, so that is free of contaminants, and this biological material then can be utilized for characterization in terms of its physical and chemical nature. A typical cell contains thousands of different kinds of molecules, and many of them may be closely resembling in physical and chemical properties. So, it becomes a challenging task to obtain a homogeneous solution of a particular substance. So, if you see, many times these molecules of interest which needs to be investigated, they exist in very small amount, that is, they might be like 0.1 or maybe less than of the total cell content. And another problem which comes in is that they may be unstable actually. So, when a substance is present in a very small amount, and this has to be brought to a purity level of almost above or around 98%, and you have to have substantial quantity for analysis or characterization. So, the task of purifying biological molecules is quite challenging, and in modern times it relies heavily on chromatographic methods. Now, these chromatographic methods started with paper chromatography. So, these include paper chromatography, then thin layer chromatography came, and in nowadays column chromatography is widely used. Now, column chromatography in particular is the most prevalent method in biochemistry for purification, and also in certain like clinical applications and other areas of biotechnology. It is a continuous separation process where separated components can be collected into separate fractions for analysis. Now, high performance liquid chromatography has revolutionized the technique where a very high degree of purity could be achieved. So, column chromatography is one where we can use it for getting a good amount of pure compound to be analyzed. Now, let us introduce little bit chromatography and let us get into a bit of history of chromatography. Now, chromatography, it comes from Greek, where chroma means color and graphene means to write. So, it is like color writing. So, it is usually introduced as a technique for separating and identifying or we can say or identifying the components of a mixture. Now, chromatography, literally meaning color writing, was first employed by a Russian scientist Mikhail Svet in 1900, and he continued to work with chromatography in the first decade of 20th century, and primarily for the separation of plant pigments such as chlorophyll, carotenes, xanthophylls and so on. That is how the chromatography started. Chromatographic techniques then developed substantially as a result of the work of two scientists, that is Martin and Singe, during the 1940s and 1950s. They established the principles and basic techniques of partition chromatography, and their work encouraged the rapid development of several chromatography methods. Some of them we are going to discuss in this section. Now, the basic principle of chromatography is that the components in a mixture have different tendencies to adsorb onto a surface or to dissolve in a solvent. Now, in all forms of chromatography methods, there will be a stationary phase. Now, this stationary phase could be solid, gel or liquid, and this is immobilized. Then there will be a mobile phase, that is it could be liquid or it could be gaseous, and it will this mobile phase flows over or through the stationary phase. So, what you have essentially is an immobilized stationary phase and a mobile or moving mobile phase which moves through the stationary phase. Now, separation of various components of a mixture is based on differential partitioning, or we can say distribution behavior between these two phases, that is stationary phase and mobile phase. So, it depends on how a particular component divides or distributes itself into the two phases. So, chromatography may be preparative or analytical. Now, the purpose of preparative chromatography is certainly to separate the component and get in a substantial amount. So, it is a form of purification, and in analytical chromatography is done normally with smaller amounts of material and is for measuring the relative proportions of analytes in a mixture, and two are both are used extensively and they are not mutually exclusive. Now, let us get into little bit the general principle of the chromatography. The basis of all forms of chromatography is partition or distribution coefficient, as we were talking about, and it is represented by Kd, that is dissociation or distribution, that is distribution coefficient. Now, this distribution coefficient describes the way in which a compound distributes or partition itself in between two immiscible phases. Now, the various components of a mixture will get separated due to varying affinities, or we can say distribution behavior of those components for the stationary phase and the mobile phase. So, if we consider two immiscible phases A and B, that could be stationary or mobile phase. Now, the value for distribution coefficient will be a constant at a given temperature and is given by the expression if you see on your screen, that is concentration in phase A, that is and divided by concentration in phase B equals the distribution coefficient or Kd value. So, this will give you a sense of how a particular analyte distributes itself between the two immiscible phases. Now, there is another term which is known as effective distribution coefficient, and it is defined as the total amount rather than concentrations of a compound present in one phase divided by the present in a phase. Now, this will be like, so here it is a concentration of a compound present in one phase divided by the total amount present in the other phase. So, it is the distribution coefficient multiplied by the ratio of the volumes of the two phases present. So, what does that mean? Rather, you are not talking about concentration as such. In effective distribution coefficient, the total amount is being taken. So, if the distribution coefficient of a compound, say for example, between the two phases is 1, that is it is equally distributed. Then, but if you have volumes between say 5 ml and 1 ml, then the concentration in two phases will be the same, but the total amount present in the two phases will be different, that is one phase will contain 5 times more than the other phase. So, these two terms kind of one signifies the distribution in terms of concentration and another signifies distribution in terms of amount of substance present. Now, there could be different combinations of stationary and mobile phases. They could be selected on the basis of a particular compound to be separated, and it should be selected so that if you are taking an analyte, it could be separated and it has a varying distribution coefficients from other analyte. So, different combinations could be, and we will be discussing these in detail as we go along, and also we are going to discuss certain techniques here, particular techniques in column chromatography based on these combinations. So, one combination could be based on partition equilibrium or partition equilibration. Now, here in partition equilibrium occurs between a stationary liquid phase and a mobile liquid phase or a gas phase. In this case, like for example, there could be a partition chromatography, there could be reversed phase chromatography, there could be gas liquid chromatography. These are the examples of partition chromatography. There could be an adsorption equilibrium. So, an adsorption equilibrium occurs between a stationary solid and a liquid mobile phase, and example of this particular type of chromatography is adsorption chromatography. Then there could be ion exchange equilibration. In an ion exchange equilibrium, it occurs between a stationary ion exchanger and a mobile electrolyte phase. So, the typical example of this is ion exchange chromatography. Then an equilibrium could occur between a liquid phase trapped inside the pores of a stationary porous structure and a mobile liquid phase, and a typical example is exclusion chromatography or gel chromatography. Another combination could be equilibration between a stationary immobilized ligand and a mobile liquid phase. So, where what has been done is a ligand has been immobilized onto the stationary phase or it acts as a stationary phase, and a mobile liquid phase passes on through it, and typical examples of this type of chromatographic techniques include affinity chromatography techniques, different types of affinity chromatographic techniques. So, you see there could be lot of different combinations of stationary and mobile phase, and these will be discussed in detail. Now, there are three modes, or rather we can say essentially two modes of chromatographic separations. Now, here are three modes where paper chromatography could be one mode, but it has been kind of replaced by thin layer chromatography. Paper chromatography and thin layer chromatography comes under planar chromatography, as the bed, or you can say the chromatographic material or bed where this separation is taking place is present on a planar surface. So, paper chromatography and thin layer chromatography bed is present on a planar surface. As far as column chromatography is concerned, the in column chromatography the stationary phase is packed in a tube or in a column. So, that is why it is called a column chromatography. So, let us little bit discuss about paper chromatography and thin layer chromatography, and then we will move on to column chromatography in particular. All right. Paper chromatography is an analytical technique essentially, and it is utilized for separation and identification of mixtures that may or may not be colored. Like earlier it started with the colored, like we were discussing about say pigments from the plants and other colored substances, but you can also use substances which are not colored, but could be stained by or could be analyzed by different methods. Now, in this technique, stationary phase is supported by the cellulose fibers of a paper sheet, and hydrated cellulose fibers, or we can say absorb water in fibers, acts as a stationary phase. The mobile phase, relatively less polar solvent system, will pass along the paper sheet either by gravity feed or by capillary action. So, if you see here that stationary phase is a paper which is cellulose fiber of a paper sheet, and this cellulose fibers or a paper could be Whatman paper. There could be modifications in this paper where you can change its property in terms of say ionic imparting ionic properties or other properties to the paper could be an option. Likewise, the water in fibers acts as a particularly acts as a stationary phase here. So, you can say that hydrated cellulose fiber acts as a stationary phase. Now, mobile phase, which is relatively less polar as compared to water, will pass along the paper sheet and will carry the mixture of substances which are present as a on the paper. So, what is done is the mixture of substances are distributed between stationary and mobile phase as per their affinities for two phases. So, substance having higher affinity for mobile phase will certainly move rapidly as compared to those having higher affinity for hydrophilic stationary phase. So, those compounds which are relatively non-polar will move in the mobile phase, but those which are not will be sticking to the stationary phase. So, this is how the separation in paper chromatography will take place. Now, here the sample or more than one sample could be placed onto the paper as a small spot or line of sample solution on a chromatography paper. I will show you in a little while the procedure here. Now, once these are spots are placed, and spots one has to be careful that how much sample is being put. You should not put too less or too high amount of sample, and it should be on a clear spot should be placed onto the paper in a, if more than one sample is there, they are in a single line. Once you have spotted and dried the paper, then it will be placed in jar containing a shallow layer of mobile phase. Remember, spots should not be dipping in the mobile phase, they should be above the mobile phase, and the jar is sealed actually. As the mobile phase rises above the stationary phase and encounters sample mixture, it carries sample with it. Now, the different components of the sample will travel different distances according to how strongly they adsorb onto the stationary phase as compared to dissolve in the mobile phase, as we have discussed earlier. Now, the compounds within the mixture will travel farther if they are non-polar as compared to polar components which will interact with hydrophobic stationary phase or surface. So, this is how a chromatogram will be run as far as paper chromatography is concerned. Once it has been run, you can take it out, and then detection of the result compounds has to be performed. So, the detection of result compounds may be performed either visually if they are colored, or by physical and chemical detection for colorless compounds. So, the detection methods may include that could be staining with certain dyes or other reagents. It could be autoradiography if you are utilizing radioisotopes. It could be fluorescence or other physical methods could be utilized. Now, after detection, the result compounds may be identified based on their retardation or retention factor, that is RF value. RF value is defined as the ratio of the distance travelled by the analyte to the distance travelled by the solvent front, and from a definite point actually. RF value will be affected by type of mobile phase, that is solvent, like say if you are utilizing different types of solvents, certainly RF value will change. The temperature, in different temperatures you will have different RF values, nature of the sample and paper will also affect the RF value. Now, let us little bit get into, like, let me show you on your screen the whole procedure here. All right. So, now in paper chromatography what is done is, like we were discussing, first thing is you have to take the proper paper, that is cellulose paper, and it could be Whatman paper. Now, this paper like I said, first thing is you have to spot your samples on this paper. This paper could be simple paper or it could be derivatized for other applications. So, what you can do is a very thin line could be drawn or you can do it visually, and here you have to spot your samples, and these spots has to be appropriate or accurate. Now, spots, more than one spot could be placed on a line here on one side of the paper. All right. Now, once you have spotted this, then this could be dried and then paper could be put in a jar, like I said, a jar containing a shallow mobile phase. Now, let us see what does that mean. So, what you have is that you have a particular kind of, say, container or a jar, there is a very simple way to make a container. So, you have a container here, and in this container what you have to have a paper here. If I can have a rod to support the paper, this is a rod to support the paper. Now, what is done is you have a paper which is hanging sort of, or it is supported paper which can remain straight in here. Now, main aim to put a rod, and this jar has to be sealed completely. So, main aim to have a rod or a support system is to keep the paper straight. Now, this paper here, as you can see, there will be spots in here as we have spotted there, and there will be a mobile face here. Now, remember, the spots are not submerged here in this one. So, what you have is, you have, this is chromatographic chamber, this is chromatographic chamber, then this is chromatographic paper which is you are utilizing here, this is mobile phase which could be a, now mobile phase mostly is, like I said, it is a relatively non-polar as compared to water. Now, so this one contains what you call, it could be, say, organic solvent water combination, and other additives could be utilized, like say it could be phenol water, it could be it could contain methanol, or lot of other different types of combinations are available. So, what you have is that once you have placed this, then automatically there will be, and this is your spots here, which are many spots are being put in here. So, once you have placed this, and this, remember, spots are not dipping in here, the water will move or the mobile phase will move up. So, this is called ascending order, or you can say it is being run in ascending fashion. There could be another way, that is, it could be, the paper could be upside down, and that will be called descending manner, where you can run that particular chromatogram also, paper chromatogram also. So, once these is as they run, these spots or the samples will be carried up in here as per the affinities for the mobile or stationary phase, and what you are going to have finally, you will be developing a chromatogram. So, finally, what you will have? You will have a paper which could be taken out from the jar, and what you will see is, you will see various spots in here as per how many spots you have loaded, and these spots could be then either if they are colored you can see them visually, or if they are not colored you can see them by staining them with different agents, or like say for example, ninhydrin is utilized to see amino acids, and there could be a lot of other different reagents, or you can use fluorescence or autoradiography to see this. So, this is about a little bit about the paper chromatography. Now, let us continue our discussion here. So, hope you have been able to understand the simple methodology of paper chromatography here. The next one, that planar chromatography system, is thin layer chromatography. Now, thin layer chromatography, this technique has almost replaced the paper chromatography with the advantages of speed, that is it could be run fast, better separations due to high resolving power. So, you have much higher resolution here, it is a much faster, and you can have a choice between different kinds of stationary phases which could be used. Now, this could be used for both analytical as well as preparative purpose. So, thin layer chromatography could be both analytical as well as preparative. Now, as we have discussed in paper chromatography, lot of things are same. So, we are not going to repeat them, but a few things about thin layer chromatography. Now, the stationary phase or the adsorbent material is mostly like silica gel or aluminum oxide or cellulose paper, and other agents are also, other adsorbent materials are also utilized. So, they will be coated thinly onto a glass or a plastic or a metal foil plate, and the mobile liquid phase will pass across the thin layer plate, held either horizontally or vertically by capillary action. Now, the process is similar to paper chromatography. Now, these plates or thin layer plates, as we say, how these are prepared. So, for preparation of these plates, layers of powdered solids in form of slurry can be applied to a clean glass plate with a binder, and then they could be dried and activated by heating. Now, TLC plates or thin layer chromatography plates are commercially available with standard particle size ranges for reproducibility. You have to have standard particle size so that you can reproduce your results. Now, they are prepared by mixing adsorbents, say silica gel or alumina, with a small amount of inert binding agent. For example, calcium sulphate or gypsum could be used with water. The mixture is spread as a thick slurry on an inert carrier sheet, and then, like I said, it could be glass, alumina or plastic. Then plate is dried and activated by heating in an oven at say 100 to 120 degrees Celsius, particularly mostly 110 degrees Celsius for 30 minutes. Now, plates of uniform thickness are obtained. Now, here for analytical purpose, the plates of 0.1 to 0.5 millimeter, and for preparative purpose 0.5 to 2 millimeter plates could be prepared. So, like I said, it could be commercially, you can obtain them without any problem as per your requirement, and you can use them. Now, TLC can be used for a lot of different applications, like paper chromatography and TLC almost have similar applications. Like in paper chromatography, it could be used for analysis of lot of different samples, right from say nucleotides, amino acids, peptides, drugs, pesticides, and in cosmetic industry, food industry, a whole lot of things, a whole lot of areas it could be utilized. Likewise, TLC can be also utilized for a lot of different applications. Now, before we go into the applications, I will just like to show you how it is done. Like I said, it is almost similar to what you call, it is almost similar to the paper chromatography. Now, here rather than a paper, what is there is a glass or say certain plate which could be plastic or thin foil. Now, on this thin foil you will have say slurry which is prepared in here, and this will be poured on this glass plate, and it will be spread evenly, which will be put in whole place, and it is like this slurry here acts as, it will have certain thickness, and this slurry will be utilized or will be acting as the stationary phase, which could be like it says silica gel or alumina gel or others. So, all other things are as the paper chromatography except for the stationary material, but it has lot of, like I said, lot of advantages over paper chromatography. All right. So, now TLC could be used for monitoring the progress of reactions. For example, it could be used for monitoring of organic chemistry reactions, say at different stages the compounds which are made could be identified by their RF values. Then identifying the compounds in a mixture or to assess the purity of the compound, that is also a very important application of both paper and thin layer chromatography, where you have a pure compound and you can assess the purity of that compound. You can use it for separating a lot of different kinds of substances or compounds. For example, you can use it to separate lipids, steroids, alkaloids, sugars, phenols, nucleotides, amino acids and peptides, and many others actually. It could be used for detection of pesticides and insecticides in food and water. It could be utilized for identification of constituents of a medicinal plant. It could be utilized in forensic science, and it could be utilized to assay the radiochemical property of radiopharmaceuticals and so on. So, there are a whole lot of different applications of thin layer chromatography. Now, practical advantage of thin layer chromatography or paper chromatography over column chromatography is that large number of samples can be studied simultaneously here, rather than in column chromatography you can use at a time one mixture, sample mixture could be analyzed. All right. So, we have discussed about two planar chromatography techniques, one paper, another is thin layer chromatography. Now, let us discuss about the column chromatography, and which is the main topic here. Now, in column chromatography, this is a separation technique in which stationary phase or stationary phase on a suitable supported matrix is packed in a column or a tube made of, it could be made of glass, metal or polypropylene, that is plastic. Now, the mobile phase passes through the column either by gravity feed or by use of pumping system, as we will discuss in coming lectures. Now, the method has become the most commonly used mode of chromatography. So, let me show you the how column looks like actually. Now, if you see the column is a simple tube like or column structure, which is closed on one side. It will be closed on both sides while running, but to support the matrix there is a filter, a disc with a filter, and the material will be filled in this here, which is your matrix. So, stationary phase or matrix is put in here. The mobile phase will be put in from top. Now, through gravity or pumping system, as I will show you in the coming lectures, and then drops which are which like comes out, and as per the affinities to the stationary mobile phase, the analyte will be separated and these fractions can be collected separately. So, this is a simple depiction of column chromatography, and we are going to see it more as we go along. All right. So, let us move on this lecture. So, what we have done is in this lecture, what we have seen is you have like different combinations of stationary phase and mobile phase, and there is a distribution coefficient, a term known as distribution coefficient, which defines how a particular analyte will separate between the two phases, that is a stationary phase and a mobile phase. Now, that depends on the affinity of that particular analyte for two phases, and the separation is based on of different analytes in a mixture in that they have different affinities for the two phases. There are different modes of chromatography systems, and there could be different combinations of a stationary and mobile phase, as we have seen adsorption, equilibration, or ion exchange equilibration, or say partition equilibration, and there are a whole lot of different combinations could be there. And the modes of chromatography, there are paper chromatography and thin layer chromatography, which are planar chromatography techniques, and then there is column chromatography, where you have a stationary phase being packed into a column. Now, before we end this lecture, let us see, as we are going to discuss more about column chromatography, a few terms, and we will continue to discuss these terms in the next section, but few terms which are important, let us discuss them here. One is chromatography. As we are listening this term, what does chromatography means here? Now, chromatography is a physical method of separation, where components to be separated are distributed between the two phases, that is stationary and mobile phase. So, that is what chromatography means. Then second term is chromatograph. Now, here if you see, chromatograph is an equipment that enables the separation in chromatography. So, chromatography is the technique, whereas chromatograph is the equipment or the instrument that enables the separation in chromatography, and that or it enables to do the chromatography. Now, there is another term called chromatogram. Now, chromatogram is the visual output of the chromatograph in the form of different peaks or patterns. Like say, when you are separating different analytes, then as they come out of the column, then they will be coming out in a particular pattern, and these are represented as peaks, as we will be showing you that in a little while in the next lecture. So, chromatograph gram is another important term here. Then stationary and mobile phase, that is the substance which is fixed in place for the chromatography method, and it is immobilized on a support, or stationary phase could also be separate and could be immobilized on a support particle and can be packed. It could be solid, gel or liquid. Then there could be mobile phase, which is the phase which moves onto the stationary phase or through the stationary phase, and then it carries the sample as per the affinities with it, so that the separation could take place. So, like here you have different ways you can, like we said, different combinations of stationary and mobile phase. Now, mobile phase is also, there are two more terms. One is called eluent. Eluent is the carrier portion of the mobile phase, and it moves the analyte in the chromatography or chromatograph, that is instrument. So, eluent is the solvent, that is part of the mobile phase, but it is the carrier portion, and eluate, another term is eluate, which is the mobile phase leaving the column, and it consists of both carrier and the analyte material. So, these two terms are important, one is eluent and another is eluate. Eluate is which is coming out of the column, eluent is the carrier portion of the mobile phase. Then analyte, analyte is the substance to be separated during chromatography. So, analyte could be, if you are assessing the purity, it could be single analyte. Analyte could be present in a mixture or a sample containing lot of different analytes, which needs to be purified. Say one analyte could be purified from a mixture, or there may be more than one analyte could be purified from the sample. So, these are few terms which are related to the chromatography, where we have discussed about chromatography, chromatograph, chromatogram, stationary phase, mobile phase and analyte. So, in the next lecture, we are going to discuss certain terms which define the performance of chromatography systems, like for example, retention time, retention volume, capacity factors, resolutions, and all different kinds of terms we are going to discuss. To summarize this lecture, what we have learnt today is in this lecture is that there is one the chromatography separation occurs due to the varying affinities of a particular analyte in the two immiscible phases, that is phase A and phase B, and their distribution is measured by distribution coefficient. That is, if say distribution coefficient is 1, it means they have distributed themselves equally. Then there is the effective distribution coefficient, that is the amount in the two phases rather than the concentration in two phases. There could be lot of different kinds of combinations, and stationary phase is mostly solid or liquid, whereas mobile phase is either liquid or gas. There could be different modes of chromatographic systems like paper, thin layer and column chromatography. And our discussion in the next lecture is going to be on basic concepts relating to performance of the chromatographic system and various techniques about the column chromatography. Thank you.